اسأل تبع الميكاترونكس.
Jadi mulai Pak Ali? Ya, baik. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Honorable Dean of Faculty of Animal Science, Universitas Gadjah Mada, Professor Ali Agus, and also the President Director of PT HRL International, and also all of the participants. It is such a great opportunity for us to have Professor Tegran here to share about immunology and its related problem. And for the first chance, I would like to invite Professor Ali Agus to give an opening remark. Professor Ali Agus, time is yours.
Baik, terima kasih Bapak sekalian. Selamat siang, salam sejahtera bagi kita semua. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, Professor Tegran, Dr. Haik, Dr. Aktif, and Dr. Sandro, and team, also Pak Heru, dan Bapak sekalian. First of all, I would like to express our sincere thanks for your coming. And this is a good opportunity for us at least to know more or less about the immunology in relation with several disease, especially HIV after AIDS atau HIV dan SIDA. Ibu sekalian, mungkin sekedar informasi terlebih dahulu apa korelasinya pertemuan kita ini dengan tantangan-tantangan berikutnya. Dan kebetulan yang hadir ini divers, ada Pak Heru ini yang aktif juga konservasi plasma nutfah, kemudian produk-produk herbal, lakukan penelitian-penelitian sampai untuk membantu kesehatan manusia. Hubungan antara pangan dan kesehatan sangat dekat. Ya. Demikian juga dampak-dampak lainnya, termasuk fungsi-fungsi kekebalan tubuh. Nah, HIV juga demikian. Ya, sehingga bagaimana mengaitkan antara pangan, ya, kesehatan melalui peningkatan fungsi kekebalan tubuh sampai mitigasi termasuk HIV. Kalau Ibu sekalian memperoleh informasi yang akurat, mungkin agak khawatir dan bisa jadi grogi betapa perkembangan dan pertumbuhan HIV di Indonesia ini sangat cepat. Bahkan di Jogja saja, kalau data resmi sekitar 4.000 pasien. Tapi kalau yang tidak resmi hampir 7.000. Ada beberapa anak-anak kecil, ya, termasuk di Solo, di Jogja. Di Solo itu kami sudah berkunjung di beberapa tempat. Ya, itu ada 170-an orang yang menjadi yatim dan piatu karena HIV. Itu aja diusir-usir, ya. Di Jogja juga demikian, tidak sedikit. Karena kita nggak ngelihat ini orang positif HIV atau tidak, itu nggak nampak. Nah, kebetulan sejak setahun lebih ini kami mencoba melakukan kerjasama, ya, nanti berawal dari pakan, kesehatan ternak, kemudian produk hasil ternak, kemudian syukur sampai ke functional food untuk mitigasi, meningkatkan fungsi kekebalan tubuh dan kesehatan konsumen. Ini relevan dengan jihad kedaulatan pangan. Jadi dari pangan, functional food of animal product, ya, sampai ke kesehatan, maka kita akan melakukan kerjasama. Mudah-mudahan 2 tahun ke depan itu kita bisa ada produk yang bisa diandalkan, dibanggakan melalui kerjasama ini multidisiplin. Ya. Kalau Pak Heru dengan timnya itu agak pendekatan-pendekatan ya herbal dan energi atau apa, sudah ada klinik yang sedang dikaji, sudah dibuka, ya, di samping peternakan. Kemudian kita bekerja sama dengan Profesor Tegran ini dari Armenia yang banyak melakukan penelitian-penelitian terkait dengan virologi, imunologi, dan sebagainya. Nah, tentu secara tidak langsung ilmunya penting bagi kita di peternakan untuk mendukung gagasan yang pernah saya sampaikan tentang bengkel ternak. Jadi setahun ini kita sudah uji coba melalui pengembangan feed supplement dan aditif ya untuk membantu fungsi kekebalan tubuh ternak supaya ternaknya sehat, produktif, pangan hasil ternak juga sehat, syukur menyehatkan. Itu kira-kira korelasinya, koneksinya ya. Sehingga kita banyak PR yang bisa kita kerjakan, terutama untuk mengantisipasi antibiotik free untuk ternak. Gimana caranya? Solusinya kayak apa? Ya. Nah, siang hari ini kita berterima kasih Profesor Tegran dari Armenia, salah satu senior researcher dan scientist di Academy of Science. Ya. Bidangnya virologi, dan memang ini mungkin kita sambil nunggu kawan-kawan dari kedokteran ini ya. Karena ini partnership antara ya dari peternakan terus kedokteran, dari nutrisi manusia, kita coba koneksikan nanti. Sehingga kenapa kita beberapa tahun yang lalu mengembangkan lebah madu dan sebagainya ini. Ya mungkin kalau kita tidak mengikuti ini bisa dianggap ini zigzag, ini kemana ini larinya ini. Padahal kita ingin tujuan akhir dari produk pangan adalah untuk kesehatan konsumen. Syukur bisa free antibiotik, itulah PR kita. Nah, siang hari ini Profesor Tegran akan menyampaikan gambaran ya, terkait dengan bagaimana ini apa tantangan-tantangan korelasi kekebalan tubuh sama HIV dan AIDS, dan setiap orang punya potensi, punya risiko ya. Meskipun yang paling ringan ya, paling ringan itu adalah ketika fungsi kekebalan tubuh terganggu itu adalah mulai dari mekanisme alergik, ketika alergik nanti mudah kena ini ya, flu, demam dan sebagainya. Dan ketika ini sampai parah, fungsi kekebalan tubuh terganggu sampai disfungsi, itulah akhirnya AIDS dan HIV. Ya. Itu yang dan berbagai virus belum ada obatnya. Ya, nanti virus yang paling lemah sampai paling kuat, yang paling ganas. Ya. Dan ini menjadi tantangan dunia, tidak hanya kita, dan Indonesia tidak sedikit pasien yang menghadapi. Ya, kalau kita ikuti beberapa ini, entah itu termasuk cancer ya, cancer di samping ini imunologi dan cancer. Nah, penyakit-penyakit lain akan mudah masuk ya, ketika fungsi kekebalan tubuhnya menurun dan berkurang. Jadi kita berterima kasih, bersyukur ya siang hari ini mungkin waktunya tidak terlalu lama, kita akan coba melihat ya dari sisi basic science-nya, ya, imunologi dan keterkaitannya. So I would like to express thank you Professor Tegran. You will give your expertise about humans immune deficiency provoked by virus and so on, how mode of action and so on. So our audience will follow and catch up the information. And this we will be more or less familiar with these issues. So once again, thank you very much. Thank you, Pak Haik and the team, Pak Heru. Terima kasih dan selamat enjoy the lecture. Thank you. Please, time is yours, Pak.
Thank you very much, Professor. It is a pleasure to be here among of you, see all of you. Thank you very much for your interest. Thank you very much, professors. And thank you, team, for organizing this meeting and seminar. It is a very big opportunity to discuss some issues, very important issues on HIV and AIDS problem. I am from Armenia. I am professor in a field of immunology and virology. So I study HIV for more than 10 years. I started to work with the laboratory of one of the discoverers of the virus in France, the laboratory of Professor Luc Montagnier. If you know, he's a Nobel Prize winner and discovered first time the HIV virus. So I started working with professor in Paris laboratory, and then I involved in HIV and AIDS clinical trials and immunology, first in Armenia, Russia, and then in African countries. So in Congo, Zimbabwe, Zambia, and then continue to work in India. So I'm very glad to meet all of you here. And let me start from the problem of AIDS and HIV. The prof asked me to uh, uh, discuss with you some immunological problems and virological problems, up-to-date problems we have in the field. But let me start from the some medical points about treatment of HIV and AIDS. And then if we will have a time, and if you will not be tired, I will go to modern problem on HIV virology, HIV immunology, pathology, and I will glad to answer all of questions. So, what is AIDS? It's a disease, but that caused by human immune deficiency virus. There are two types of virus: the HIV-1, HIV-2. The viruses affect the immune system of the body and exposing to the body to a number of disease and infection. This is a classical explanation what is AIDS and what is the HIV virus. The picture, this is small picture of HIV. You show, you see here. So, what is the current anti-HIV therapy? It's using of so-called medicines, antiretroviral drugs. The goal of these drugs is treatment disease to suppress the viral replication. Everybody know what is a replication. This is multiplication of virus inside of cells. So the goal is the treatment is suppress the viral replication as much as possible and as long as it's possible. At present, this requires combination therapy. What is the combination therapy? I will explain here from the next slides. The slides show the life cycle of HIV and potential targets, drug targets. If you see, there are the many stage of HIV infection starting from the first point when the virus meets the target cells. And for the HIV virus, the target cells is a CD4 T cells. So at the beginning, there is a binding or fusion of the virus with the cell target cells. And then the next step is a virus entry into the cells. After that, the cascade of biochemical events take place. The most important stage is formation of DNA from the RNA molecule of the virus. Why we call RNA and retrovirus? Because HIV virus is RNA containing virus. So for the life cycle, for the multiplication of virus inside of cells, we need to make a DNA molecule from the viral RNA. So this is very key biochemical stage. And there is one very important enzyme in the virus, so-called reverse transcriptase, that make DNA molecules from the virus RNA. So after that, some very complicated biochemical and molecular mechanisms take place, and the viral become integrated into our genome, into the genome of the target cells, and viral become integrate within the genome. So the last stages are viral maturation and new viral particles production. So each these steps are a potential targets for drug therapy. We will speak about this now. The antiretroviral drugs, there are agents developed so far after different stages of the life cycle HIV virus. One that blocks the binding of the virus to the target cells is clear. If you block the binding, virus cannot enter into the cells and we can't treat the infection. The next kind of drugs are the blockers of RNA cleavage. That means that if you can keep virus in virgin stage, in non-mature stage, you can stop the infection process. Other drugs inhibits enzyme which we call reverse transcriptase. If you remember, this enzyme making RNA, DNA from the RNA. Okay. The other drugs inhibit enzyme and virus incorporation into the cell chromosome. That type of drugs called blockers of virus integration. There are also other steps, but until now we have potential targets, but till now there are still no any medicines blocking the other stage of viral infection. So what we have now, we have three classes of antiviral drugs that are nucleoside analogs of the reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors, and so-called protease inhibitors. That's a generic name and of the drugs. What is the problem? The problem using antiretroviral drugs are these drugs should be used in combination mode. The patients need to take the medicine every day for whole life period. But the problem exists. During several last years to use combination therapies, HIV enzyme showed considerable antiretroviral activity and suppressed viral replication to undetectable level. But HIV is still slow produced and stored in different tissues. And what is the second problem? That many patients are developed to virological failure because of incomplete viral suppression. Or what is the most important thing, that there are drug-resistant viral strains development. The next problem that current therapy does not prevent HIV-mediated immune damage and does not increase a host immune response against the virus. This is the main problems. Also, there is toxicity. There is several side effects for all of class of antiretroviral drugs. So, every scientist working on HIV and AIDS want to make a miracle medicine, a new one that can cure HIV infection. So we have generated some ideas that about what ideal antiretroviral drug should be. It should be effective against HIV-1 and HIV-2. It should be reduced viral production from infected cells. It's clear. It should possess broad spectrum antiviral, antibacterial, antifungal activity. It's also clear, yeah? But what is very important, the new drugs should not develop a resistance. Is it possible or not? We will discuss it later on. It should be improve the quality of life, because antiretroviral therapy we use now, they reduce the quality of life because of very hard side effects. Okay, so also the medicine should be affect HIV mediated immune damage, and it should be non-toxic. Is it possible or not? Let's go on and thinking together. Maybe it is possible. So, development of new class of antiretroviral compounds with a different mechanism of action on viruses, bacteria, and immune system is a very important goal. So let's speak about natural essential oils as a new anti-HIV and AIDS drug. The optimal management of the infection is very difficult objectives to achieve. Well, there are many therapies available. Current drugs have much room for the improvement, particularly in the area of resistance, toxicity, adherence, and drug interactions. So the comprehensive research and development program, natural essential oils, and the company that Mr. Haik and team is presented is committed to expanding the treatment option available with powerful new anti-HIV/AIDS agents that help to address many barriers to optimal HIV treatment. What is the idea? The idea was to create a drug being versatile, destroying as many pathogenic microorganisms as possible. This is one. Having low toxicity, this is two. Compatibility with the living organism is three. And which could recognize the island in the living organism, similar to the killer cells. If you know, in our body, we have some type of cells, we call them killer cells or natural killer cells. So this is what the original idea, how should be the new medicine. But there was a new with this idea that the very interesting physical chemical model of the essential oils is based on the blood of mammals, including human blood, and the stereochemistry of moving equilibrium systems of dynamically balanced organic compounds, conformation complexity with biologically active molecules. It's a very difficult to understand, but I will show you very simple. If you see, this is a structure of natural oils and our blood. Colloid in blood, we have colloid polymer solution with water as a solvent. Colloid polymer in oil with water as a solvent is the same. Salts and low molecular weight organic compounds in the solid matter in the blood, and just like this we have in oil. So we have two compounds, the blood and oil, that are very similar, and we can compatible. They are compatible with each other. So what is the chemistry of these natural essential oils and the mechanism of action? That consists of both the direct and indirect action on each of the components, and they are working as a whole one. So what we know about these compounds, that they are exhibited pronounced antimicrobial activity with wide spectrum of action against DNA and RNA viruses, including HIV-1 and 2, as well as gram-positive and gram-negative bacteria. We have a very big experimental material suggesting that. So natural oils also displays fungocidal, protozoidal, cysticidal activity, being active even against spores. Wide spectrum of antibacterial and antiviral action. Insignificant variation in effective doses, which is not dependent on bacterial and virus species. Possibility of short-term treatment and ruling out the adaptation of viruses and emergency of drug resistance is true. All of experimental data bodies suggest these results. So, also what is very important, the lack of toxicity, insignificant side effects, lack of cumulative action, yes, which will give us opportunity for long time using of the oil components. There are also some very important characteristics, including direct transport of drug in the body. So, let's go on a possible mechanism of action of oil components against HIV and HIV-1 and 2. We suppose that all components we have in essential body, essential oil compounds can suppress all stage of virus life cycle in the target cells. So, in conclusion, I will show you some recently obtained results in my laboratory and other laboratories that we have some antiretroviral activity of natural oils against viral strains and clinical isolates, and which is demonstrated but in a number of cultivated T cells lines. Also, we have some results that show effectively that natural essential oils effectively fights to a lot of viruses, including influenza and herpes simplex group of viruses. Also displays bactericidal activity against even an antibiotic resistance bacterial strains, which are Salmonella, Typhimurium, Vibrio cholera, Pseudomonas, and Staphylococcus escherichia coli. Also, our recent results show that the essential oils possess immune modulating activity, like inducing early activation of lymphocytes, phagocytosis cells, respiratory part of the cells, upregulation of the some cytokines production, proliferation of cells, and process also which is very important, anti-inflammatory activity. So, in conclusion, experiments on various species of laboratory animals, also mice, rats, rabbits, dogs, and you can continue, yes? And as well as in healthy volunteers have demonstrated that oil is practically not accumulated in a body, and it has a very low toxicity and oral, maybe intravenous or intraperitoneal administration, maybe. So, in therapeutic and much higher doses, oil does not affect behavior, body weight, temperature, food, and water consumption. It also has no adverse effect on liver, kidney, and central nervous system. It's true. So, this is a summary of our proposed mechanism of action on HIV infection. And we will suggest to carry some clinical trials, which we'll discuss tomorrow in more details. But the clinical trials of oils should be evaluated in combination with the antiretroviral drugs in HIV positive patients using surrogate and endpoint markers, which includes study of drug effects on HIV, on CD4 cells, and on quality of life, and other adverse effects. So thank you very much. This is a short introduction about HIV and AIDS, and the new potential anti-HIV natural oil drugs the company produce for a clinical trials, which we hope will start together in a clinic. And the details of the protocol we will maybe discuss tomorrow in more details. So now I am ready to introduce you some new scientific data about immunology, about virology, and about pathology of HIV infection, if you're interested. Prof.
Okay.
Okay?
Okay, thank you very much.
Thank you. I need some something to write down to show how is the problem is big, how is problem is huge to solve the HIV and AIDS problem. We can divide this problem into three big groups: AIDS, HIV, first part is virology, the second is immunology, the next one is pathology. What we know about HIV virus, I told you briefly. Point A, that is a RNA virus. It's true? Okay. B. This virus has a very important enzyme inside, which we call reverse transcriptase. This makes RNA, DNA from the RNA. Anybody knows how this enzyme is work? No? Okay, I will explain for you. So, so the enzyme using RNA as a matrix to transfer the information from RNA to DNA. That is what is very interesting, that this enzyme working not so correct, and each time the enzyme makes some mistakes and errors. So the second problem is that, next problem is that, if the cells infected by a virus, let me say HIV version 1, this is original virus came during the infection first time. But because of working this enzyme, and the end, during maybe some 10,000 life replication cycle, we have finally HIV versions. So during HIV infections, we have not only one type of virus. We have very big cloud of virus. So as in the cloud, there are too many different viruses with different faces. So we will speak about in the immunology point. But just now it is very important to understand that the mutation rate of the HIV virus is so big, so high. So can you imagine if the rate of mutation of our somatic cells is around 1 cell per million cells. So the mutation rate of the virus is 1. So even each cell, cells from 100 cells that can mutate it. You can understand it, which is a big difference of mutation rate. So this is not finished virological part, because we have also a very big problem here. The viruses use receptor mechanism and co-receptor mechanism. What does it mean? Receptor means that virus can enter into cells via a very big gate. In immunological point of view, we call this gate CD4 receptor. We'll speak about in this part. But even this gate is closed, HIV virus can using windows, small windows, going inside of cells. So this is a very important to understand how we can block the virus and target cells interaction with and entry process of the virus into the cells. We can never close all doors and windows because our cell will die. So this virus using all small holes, all opportunities to enter the cell, enter into the cell. This is a very big problem. Okay. Also, the next problem is that HIV virus infected not only CD4 T cells, but the virus infect also progenitor cells, progenitors that are located in our bone marrow and making blood cells every time. Okay, so this is not only huge entry opportunities, but also there are many types of cells that virus can infect. So this is virological part. So what we have in immunology part. I'm sorry. In immunological point of view, we have well-known target cells that called CD4 cells. Anybody knows what is a CD4 T cells? Our immune systems like a balance. So-called CD4 T cells, there are helper cells. There are help for inducing immune response, for generation of immune response, for formation of immune response. They push our other cells to making immunoglobulins, defense mechanisms. So there are helper cells, help. But in other hand, we have suppressor cells that restrict the activity of these cells to make them in a balance. Okay? A balance. So in normal state, we have CD4 cells, which are helpers, and CD8 cells, which are suppressors, in a normal balance. But when HIV infected CD4 cells, the balance is changed. And we have small help and big suppressor. Yeah. So if we have suppression, the opportunistic infection will come very faster, very bigger. So this is one point of immune deficiency. So the number of CD4 cells dramatically decreased after HIV infection. So the goal of anti-HIV treatment is increase the number of CD4 cells. Yeah, this is true. This is clear. Yeah, but this is not only one problem. We have a second problem. CD8 cells, there are different types of CD8 cells. There are killers, and suppressors. What does it mean, killers? The killers, there are specific type of cells that can recognize in our body infected cells and kill them. It is very interesting that HIV also that part of CD8 cells that's responsible for the killing of the infected cells. So only suppressor cells, the number of suppressor cells are rising. So this is also a very big problem. In every point, I can put one such positive signal that can our national oil drug be effective. So, for example, in killing, we can stop the potential of virus kill the killer cells. How? We can push the proliferation and the multiplication of CD8 cells because of activation so-called interleukin-2 growth factors, concentration of this growth factor for CD8 cells. Our study shows that oil increase the concentration of this cytokine, and the killer are become more and more active in a body. Also, the second problem in immunology is that HIV causes uncontrolled inflammatory response. You know what is inflammatory response? Everyone is know that what is the inflammatory response. So, but during HIV infection, the inflammatory response is uncontrolled because the key cells are absent. Helpers and suppressors are too high. There is very big balance. So in organism, in our body, during HIV, we have inflammatory response. Yes. Very high potential inflammatory response. This is the immunology part. And also we have pathology. What does this mean, pathology? That HIV and AIDS is affect central nervous system, endocrine system, and immune system. The question is that which system is first affected? Everybody thinking that is an immune system. No. HIV infection causes damage in central nervous systems. After, in an early stage of infection, we have losing our neuronal stem cells. There is there are no any new stem cells in the central nervous system. So we have serious damage in the nervous system, central nervous system. The second is endocrine system. We have damaging stress, general stress response system, which is a hypothalamus, hypophysis, adrenal axis. Yeah? And finally, how we can treat HIV and AIDS? Killing only virus or improve all of these steps? This is a very big question. Until now, doctors, scientists, immunologists, working and continue to working, but the answer is still question mark. Yes, but we hope that new medicine from the company will help us to solve some main issues of this problem. Because essential oils is well known that affect and help neuronal activity, endocrine system activity, improve immune response. So, thank you very much. If you have any question, please.
Thank you very much for very nice explanation. Jadi untuk Bapak sekalian yang mungkin kuliah imunologi atau virologi, ya, karena mungkin sebagian kecuali dari kedokteran, ya, ada beberapa Bapak sekalian dari kedokteran, tim HIV-nya UGM, ada Mbak Yunri, Mbak Emi, ya, Aini, lupa Aini dan kawan-kawan. Timnya Pak Heru. Jadi kalau saintis peneliti bisa menyelesaikan persoalan HIV atau AIDS, akan bisa membantu menyelesaikan sebagian besar jenis penyakit. Karena mekanisme bagaimana virus HIV ini menyerang, ya, ternyata virus HIV ini setiap waktu mutasinya begitu cepat sehingga virus HIV itu tidak tunggal, tidak satu tipe, tetapi ini multi, ini jadi kayak cloud, kayak apa ya, gerombolan ya, dia kayak pasukan tempur yang begitu banyak, ya, kayak mega lah ya, mega besar yang mau turun hujan, kok mega, ya, mendung ya, cloud. Jadi mendung, ya, virusnya macam-macam, jenisnya macam-macam, dan dia bisa masuk dari berbagai pintu, lewat pintu utama atau jendela atau apa saja, ya. Nah, itu sehingga tidak sangat mudah. Sehingga sekali seseorang itu kena, maka relatif berat. Karena ada mekanisme lain. Di samping secara patologi juga akan mempengaruhi efeknya terhadap ini tadi, ya, central nervous system, kemudian hormonal, endokrinologi, dan imunologi. Nah, secara imunologi, virus ini juga mengganggu sistem kekebalan tubuh lewat gangguan keseimbangan, ya, entah itu CD4 atau CD8, ya, sel pembunuh itu, ya, itu terganggu. Yang terjadi adalah ketika fungsi kekebalan tubuh itu menurun, maka penyakit-penyakit lain akan masuk semua. Entah itu mulai penyakit kulit, penyakit tuberculosis, penyakit yang lain dan sebagainya. Ya. Nah, tadi ada sesuatu yang menarik bagi kita, ya, saya kira mungkin aplikasinya bisa lewat ternak dulu atau hewan, adalah pemanfaatan essential oil, natural essential oil yang mulai sejak saya kira hampir 15-20 tahun terakhir ini banyak dieksplorasi, ya, penelitian-penelitian pemanfaatan natural essential oil. Indonesia ini adalah mega biodiversity, ya, jadi sumber-sumbernya saya kira banyak. Hanya mungkin penelitian ke arah situ belum tertata dengan baik. Ya, kita punya mega biodiversity tapi tidak begitu optimal. Kita ya belum ngerti nih obat mana yang paling efektif. Nah, mudah-mudahan dengan partnership yang sedang kita rancang, kita bangun, mudah-mudahan bermanfaat tidak hanya Indonesia, mungkin dari kita. Ini ada timnya Pak Profesor Bavikian, Haik, Sandro, Rajib, yang sejak hampir 4-5 tahun ini mengaplikasikan paket teknologi ini, memanfaatkan natural essential oil Ya sebenarnya tidak semata-mata natural essential oil, ada oil yang diformulasikan yang ditreat ini untuk mengatasi virus pada udang, ya. Sehingga udang yang 3 tahun yang lalu gagal panen tidak bisa diekspor, ya karena ada virus, what's the name virus in shrimp? Ya karena kita bukan orang perikanan, jadi karena kita orang perikiri. Ya, kalau perikanan kan ikan, kalau perikiri itu hewan. Jadi kita orang perikiri. Nanti akan dijelaskan sedikit Pak Rajib bahwa virus yang menyerang udang itu luar biasa. Mungkin bisa jadi itu juga HIV-nya pada udang. Ya. Habis semua ya karena kekebalan tubuhnya terganggu dan akhirnya banyak gagal industri tambak udang. Tapi sejak beberapa tahun terakhir ini sudah relatif sukses ya sehingga mungkin ada masalah-masalah kecil tapi secara umum sudah sukses. Tambak udang bisa berkembang lagi, bahkan Indonesia menjadi salah satu eksportir terbesar ya udang. Nah, kemudian aplikasi dari teknologi ini setelah di peri kanan mau masuk ke peri kiri, ke peternakan ya, di babi, di ini pre-trial kami sudah melakukan di beberapa. Nah, dari pre-trial ini ada produk, entah itu kalau sapi perah nanti susunya, kemudian kalau ayam, petelur atau etek kita sudah ininya mudah-mudahan punya properti yang sama itu bisa digunakan untuk membantu mitigasi berbagai penyakit, salah satunya HIV. Jadi ini loh Bapak sekalian backgroundnya. Ya nanti si BC akan teruskan penelitian pada sapi ya, bagaimana membengkel pedet, jadi pedet yang kurus-kurus, gudikan, penyakitan dan sebagainya itu kalau fungsi kekebalan tubuhnya nanti bisa masuk ya, jadi kamu bisa melihat ini bagaimana keterkaitannya itu menjadi memudahkan landasan teori berpikirnya ya. Ini memang termasuk pengetahuan yang agak mutakhir, terkini dan sangat relevan dengan kondisi kita di Indonesia ya, karena kita mega biodiversity herbal. Ya, kita ini kan minum jamu, ya, misalnya minyak dan sebagainya. Jangan-jangan kalau minyak angin itu minyak apa yang serba minyak-minyak itu diminum aja malah membantu kekebalan tubuh. Tapi perlu diuji, dicek dan sebagainya. So thank you very much. I just explaining in Indonesia, in bahasa Indonesia a little bit.
If you have any question, please.
Jadi kalau ada pertanyaan, silahkan. Kita besok akan lanjutkan diskusi karena untuk menyusun protokol penelitian yang lebih komprehensif, pemanfaatan natural essential oil ya, natural essential oil yang sudah diformulasikan oleh timnya Pak Haik, Rajib dan sebagainya untuk is there any question? Kalau ada pertanyaan silahkan. Or maybe you will give additional information, Haik or Rajib or Andrew. Do you want to add some information? Oh, Haik. Dr. Haik is pharmacist. Jadi penelitian multidisiplin sekarang sangat dibutuhkan. Ya. Kalau melihat ini itu sudah multidisiplin, tidak mungkin kita bisa ini. Ya. Supaya kita agak nyebrang-nyebrang sedikit dapat ilmu lain. Monggo, please, Dr. Haik.
Thank you for the opportunity given to us and for Prof. Tegran to express part of his knowledge and his experience in managing and treating HIV patients. What I can add is it's part of our commitment to find natural solutions for different diseases, and we know that HIV is a sensitive topic to all countries because it's somehow it gives a stigma on the patient, so he cannot practice his normal life. So us by developing such formula that has a very high safety profile, and as we demonstrated in some trials that it has also very potent efficacy as an antiviral property and as immune modulating efficacy. So our formula is based on natural essential oils that are all safe to use, human food grade. So safety of the patients that are using it is utmost priority for us. And of course that we want, and the medical society want innovation solutions for difficult diseases. So that's what why we are here together with this prestigious university to help give more hope to patients with HIV. So I think tomorrow we'll have also fruitful discussion and collaboration on the protocol that will be discussed, and hopefully we can start this project as soon as possible. Thank you.
Okay, thank you, Dr. Haik. Pak Rashid, would you on shrimp, little bit information on shrimp, and then how to...
Yeah, thanks, Prof. Ali. So in addition, what I would like to say that, we have been working on shrimp diseases with Prof. Babikian and Dr. Haik and his team for last 4 or 5 years. And we have been able to find solution for white spot syndrome virus, infectious immunomycosis virus, which is IMNV. It's the same RNA virus as AIDS. And we also work on some bacterial diseases of shrimp and fish. So I hope we will have the same success working with human here. Okay. Thank you very much for the opportunity.
Okay, thank you, Dr. Rashid. Monggo, kalau ada pertanyaan, Mbak Aini, Yunri, Mbak Yunri, kalau enggak nanti besok kita akan diskusi intensif, tapi kalau ada informasi tambahan. Some of them are master students, some lecturers, and so on. Ini informasi dari Pak Heru. Kita tanya Mbah Google lewat berita ini. Berdasarkan data dari PBB, untuk HIV dan AIDS atau UNAIDS ya, di Indonesia terdapat berapa jumlah pasien? 690.000 penduduk yang telah terinfeksi HIV. 690.000 HIV. Ya. Namun dari jumlah ini, angka orang yang mengetahui status HIV-nya masih kecil sekali, yaitu baru sekitar 30%. Dari angka estimasi tersebut, Kementerian Kesehatan juga menunjukkan bahwa jumlah ODA atau orang dengan HIV atau AIDS yang saat ini menerima terapi pengobatan antiretroviral baru sekitar 8%. Kalau 8% itu baru sekitar 65.000. Ini adalah angka yang paling kecil di antara negara-negara region Asia Pasifik. Jadi ini sebenarnya awareness bagi kita semua ya, bahwa HIV ini menjadi momok paling besar, sehingga kenapa ini sebenarnya Presiden Jokowi beserta tim berharap kegiatan ini bermakna untuk membantu. Yang paling simpel ya, kalau memungkinkan ini samping lewat natural essential oil, itu adalah lewat produk-produk peternakan. Ya, jadi essential oilnya kita lewatkan ternak, nanti lewat sapi perah atau lewat ayam petelur atau itik petelur, nanti lewat produk inilah. Itu yang secara masif mungkin akan murah. Kalau ini terjadi, mungkin bisa menjadi sebuah revolusi. Ya, kalau ini bisa tercipta. Ya. Ini informasi tambahan. Jadi ini masalah serius karena kesehatan itu ongkosnya mahal kalau sampai terjadi berbagai penyakit. Karena penyakit yang ada di Indonesia itu penyakit tidak menular ya. Itu konon jumlahnya 30%. 30% itu kalau jumlah penduduk Indonesia 250 juta, 30%-nya berarti hampir berapa itu? 80 juta. 80 juta orang Indonesia itu mengidap penyakit tidak menular. Ciri-cirinya tekanan darah tinggi, ya, kolesterol tinggi, nanti stroke, kanker, gagal ginjal karena glisemi tinggi, termasuk HIV dan penyakit-penyakit lainnya.
Can I ask a question for Prof?
You want to ask the question?
Yeah,
please.
Hello, Prof. I have a question for you.
Yes.
Since we are in the in a very high technology world now,
Yes,
an advanced technology.
yes,
And since we are be able to um, to detail the virus uh, as you detailed it,
yes.
where are the doctors are stumbling to have a cure for such a disease? Which phase they are they cannot solve? Where did they solve and where they are stumbling in the phase that they
Okay,
cannot
okay.
solve?
I will show you in that part. Okay, I will show you.
Like, is the complexity of virus or lack of
No,
knowledge?
no, no, no,
Yeah,
no, no,
is it
no.
a lack of knowledge or complexity of virus, or what's the main reason that we still don't have a
So if you see, this is a life cycle of HIV,
yes,
and also an well-known targets
yes,
that the potential targets for treating the infection, the
Mhm.
fighting with the infection. So if you see, there are too many stages
Yes.
here, but we can divide into actually 4 parts.
yes,
The first is binding, the second is viral integrase. It means that virus become a part of our genome.
okay,
So the next part is viral regulation. That means making some RNAs, DNAs, and biochemistry. And what's also very important is a maturation of the virus. So this is 4 actually my cycles. For each of them, we have no effective targets, effective medicine, effective drugs. Why?
why?
Why? Because the rate of the mutation of HIV still too high. If our body cells changed like 1 cell per million, virus can change 1 even per 10. You understand?
Yes,
But
different phases.
imagine each time per a day in one body, uh, billions virus cells, viral particles can generate it. Billions. But the number of immune cells decreasing, 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 not to thousands, even to 0. You understand?
Yes,
So the rate of the mutation is still a very big problem. So we're doing something in our lab, we synthesizing some active molecules and go to the clinic and discover that the molecule doesn't work because
it's changing.
the time we're losing from the laboratory to the clinic. There is a time, during that time, a virus become a resistant. So we late. So we go back to lab, started to make a new medicine, go to clinic again, and we lose again. We discovered that we lose again the time.
Mm.
You understand?
Yes.
Because this is a continuously process, continuously generation of a very huge diversity of the virus. This is also true for a bacteria. You know, we have now today serious problems called tuberculosis, which is a very major issue. This the bacteria become multi-drug resistant. What is mean multi-drug resistant? It means if bacteria resistant to one drug, he become a resistant to all other rest drugs. So there is chance to cure. This is the same problem. That's why we cannot produce also a vaccine, effective vaccine, because we go to lab producing one sequence of the virus, coming back to the clinic, we are again late.
And this is all the 4 cycle?
It's all 4 cycle, all the 4 cycle. Because the structure of this virus is very interesting and dynamically changed every time in each stage. And patient to patient, they are very, very different. Even in single patients, the in the very time of beginning infection and the late stage, you are too many different from the virus. Many, the difference is huge between first stage and late stage. This is a problem. Yeah, our immune body, our immune system cannot fighting against with such high diversity, and our laboratory, our molecular technologies also cannot. So we wish to find the solution from another part, using more natural components, which can working integrally. I mean, working all on steps, all on tissues, all on organ levels, and what is very important, at the level of regulatory systems. That means neuron system, endocrine system, and immune system. That is only one chance. We have no another.
What about the existing medicine now in the market? What does it do? Does it suppress the virus in your body, or what does it exactly do?
Okay, we need just now to use combination. If you remember, I show you the slides.
Yeah.
That we need to use combination therapy. That means we have to use different class of drugs, effectively blocking each every known steps.
Mm.
Okay, but these drugs still very high toxicity, and virus become resistant.
Mm.
So for antiretroviral therapy, we need maybe time by time doing some tests for patients, study the resistance profile of the virus, and changing the antiviral therapy regime each time, maybe after 2-3 months, each after 3 months, maybe 4 months, even maybe one after month, every month. So we have no so big arsenal of medicines now, clinically and officially approved medicines.
So what does it do? It blocks it so it doesn't become AIDS, right? So
Yes,
it stays
yes.
on HIV.
Yes, yes. But there is also one other possibility, you know, maybe combination of the chemical drugs with the natural drugs will help solving many problems. For example, problems of toxicity, tolerability, side effects, improving immune system, improving neural system, improving, let me say, metabolism of the patients, because many patients suffering is the suffering weight losses, dementia, neurodegenerative disorders, etc., etc. Maybe combination of well-known antiretroviral therapy with natural products, with, for example, natural essential oils, will help solving many problems. I believe it. I believe it.
One last question.
Okay.
One last question. Does your lifestyle affects the disease? Does, for example, if you smoke more or you are not,
Definitely, yes. Definitely,
does
yes.
it?
Yes, yes. But the harm from virus side and harm from the using combination therapy is you cannot compare this with the smoking or lifestyle. But if you know, HIV is also lifestyle diseases.
Yeah,
You know,
because I
because
know.
who is suffering HIV mostly? Intravenous drug users, homosexual. Yes,
Yeah,
you know.
yeah,
So everything
I know.
causes decrease of the immune balance in a
Mm.
drug user, drugs, narcotics, yeah, and also heterosexual relations that are causing a down regulation and of the activity of the immune system.
Okay. Thank you.
Thank you.
Thank you Pak Sandro. Jadi obatnya harus multi target dan sampai sekarang tidak bisa membuat vaksin atau obat yang hebat untuk ini juga sampai sekarang belum ketemu. Paling hanya menurunkan. Ini concernnya seluruh dunia ini. Ya. Mungkin apalagi kita ini belum. Kalau ada pertanyaan lagi? Oh. Prof. Tegran, is this PowerPoint presentation can be shared?
Please ask Haikan team because I have no computer, no nothing. I do it.
Okay.
I do it tonight.
We will find.
I started making PowerPoint
Yes,
presentation
definitely.
and 12 at night and finish around 4
a.m.
4 a.m. And the early morning I fly from Jakarta to Jakarta. Okay. So there are a lot of mistakes in. Maybe we first will correct it, them and
Like spelling things. We'll fine tune it
Yes.
and.
Bisa Pak Heru, nanti bisa dibagi setelah dikoreksi, mungkin ada beberapa koreksi.
Card also, I'll email it to you.
Oke, ada lagi about CD4, Professor Tegran.
Yes.
One of the indicators of the severity of the HIV patient is viral load and CD4
cells,
levels,
yes.
CD4 cells. How important the CD4 as the indicator? For example, if the treatment can improve the CD4 level from 200 to become 300, for example. And then how the minimum level of CD4 must be.
Okay. Can I draw one diagram?
Okay.
For doctors working on HIV. Okay.
Okay. Thank you. Next itu nanti langsung imunologi. Ilmu susah dan langka. Ujian nggak lulus nanti. Nanti pengujinya si Aini sama Juni.
This CD4. This axis is CD4.
Yeah.
This is viral load. This is time.
Yeah.
This is time. We have 0.0. This is the time of infection. HIV penetrates our body. At that time, we have a very high CD4 level.
How many?
It depends. For example, up to 2,000 and 2,500 per millimeter square. Okay. This is normal level. This is normal, okay? At point 0, we have very normal and very high CD4 level. After some time, which we call asymptomatic period or no AIDS period, we can divide this time on non-AIDS time and AIDS time. AIDS is mean that the syndrome is becoming and clinically diagnosed. But in this time period, we have no symptoms. We can diagnose only HIV infection by laboratory tests, looking for virus load. Okay, so what happened with CD4? So CD4 decreasing slowly to the level which is the minimal normal limitation of the normal range. According our understanding, studying huge population in European countries, Asian countries, the minimum level, let me say, this is something 450, 1250. This is a lower threshold, lower threshold for a normal fluctuation. This is a high, this is a low level. So if during a time which depends from patient to patient, Sandros asked if patient smoking, if patient drinking, something else, life cycle, this period is closer, shorter. Okay, so in this point, we have very culmination point. It is everything happens in this point. After that, maybe patient go in stable situation because of strong therapy, because of good lifestyle, because of maybe say essential oil, I don't know, is go to the stable state, or go dramatically down, even to 0. I saw patients, many patients. 40. Yes, no, 0. I saw, I saw maybe 100 and more patients living with 0 CD4. In Africa, in India, in Armenia, in Russia. I saw this patient. Even with 0. Even. This is what happens with CD4. What about viral load? You know, at this point, 0 point, viral load also very high. That means HIV infection will be in 2 different stages: acute infection and chronic infection. At the early beginning, we have acute infection, too high level of viral replication. But because of CD4 is high, the replication is go smaller and smaller down. Yeah? I'm sorry. I misread from this point. Okay? Slowly, go. And in this very critical point, goes up. Yes? This is a natural story of the HIV infection and AIDS. So the main parameters still CD4 and virus load. Why? Because they are very easy to measure. But the real picture is not a CD4. Real picture is a immune is a measuring of all immune state. That's mean CD4, CD8, and CD8 subpopulations, CD4 subpopulation, and CD19 B cells. And CD natural killer cells. This is real picture, but it is very expensive. So we can reduce on a level only CD4. Keep this in mind.
Yeah.
This diagram.
It's very important. Basic knowledge.
It is not a basic knowledge. It's a basic clinical knowledge. Yeah. Basic knowledge is how the virus is escaped from the immune response. It's a critical. So it's have to keep in mind this, and for each patient you may operate individually.
Great, thank you, Prof. Tegran. Mbak ini yang dari kedokteran. Ini timnya perlu Thanks. Thank you very much. It's a very nice presentation, understanding much better for us. And tomorrow we'll continue. Bu Yandi bisa ikut ya? Katanya Rabu bisa.
Thank you very much for your interest. Hope tomorrow we will discuss more details on a protocol. Yes.
I revise my statement. 300 not only from from Puskesmas Sidang and also Puskesmas.
From Yogyakarta ya?
Yeah,
So,
Yogyakarta.
yeah.
Almost every one day, every one per day.
one per day.
Wow.
Only one.
Okay, thank you very much. We'll continue and give a big hands to our. Yeah, take it.
We need the souvenir, Prof. Ali Agus, for Professor Tegran. We would like to give you a souvenir.